the screen is nothing. Ah, okay. Ah, yeah. Okay. Okay, I'll just go here. Oops.
It's just like in the change, since we were all coming from warm places. It's just the sensation. It was like weeks that I didn't have to put something on the outside. Yeah. 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 TVTs and no, I think it's more in the sense like bars or like we went to the one bar and it was like it's a night like this and like that. It's just
It doesn't matter. Good morning. Welcome everybody to this symposium about non-invasive neuromodulation of deep brain structures to enhance cognitive functions. Well, great that you made it so early, even the third, fourth day of the meeting, so it's becoming more and more difficult to come in the morning, I guess. For cognitive function, it's quite clear that these functions like spatial navigation, like motor learning, or like episodic memory, they are represented in larger networks. And in these networks, there are also like deeper structures, subcortical structures, like the hippocampus, for example, the hippocampal entorhinal complex, or the striatum. And so far, with non-invasive methods, due to the kind of steep focality depth trade-off of other non-invasive brain stimulation methods like TMS or TDC, TAC, STR, and S. So these deep structures were basically not reachable with these methods without basically stimulating a large part of the cortical areas above or more close to the skull. So today in this session, which we invite you together with the co-chairs Olaf Blanke and Neil Grossman. They can unfortunately not be here in person today. We invite you to 3 talks where there will be presented first data and new approaches, how probably these deep structures can be reached and can modulate the activity of these structures, and also influence the respective behavior and activity in the brain. So we have basically 3 talks. I mean, maybe go quick to the other part. So we have 3 speakers. Ines Violante will start with her talk about non-invasive temporal interference electric stimulation of the human hippocampus. And it's followed by the presentation of Elena Beanato, modulation of human striatal activity with temporal interference stimulation. And the session will be finished by Hyuk-Jun Moon, impact of MTL-targeted TI stimulation on spatial navigation, probing with grid cell-like representation. So I would like to invite Ines on the floor for her presentation. Thank you.
Thank you. Morning. Oh yeah, just pointer and everything. All right. So good morning, everyone, and thanks for coming so early. So really excited to talk to you about the work that we've been developing using temporal interference to target the human hippocampus. All right. So what's all the buzz about, what's all the hip about? So a couple of years ago, Nir showed in this paper in Cell that one could target the hippocampus. Okay. One could target the hippocampus in the mouse without eliciting activity of the underlying cortex. So what you see here is the quantification they've done in the hippocampus. So they look at CFOS activity, so neuronal activity in slices. And what they show is that they have more activity in hippocampus, that they have underlying, in the cortical regions, underlying the electrodes. In a nutshell, the excitement, it's about having a technique that will allow you to do non-invasive, deep, and focal brain stimulation. And that's something that we really want for a number of targets. So the hippocampus. In humans, the hippocampus lies within the medial temporal lobe, so deep in the brain. It's this red structure that you can see here. And the hippocampus is critical for associative learning and memory. It is also one of the very first structures that gets damaged in early stages of Alzheimer's disease. And interestingly, at early stages of AD, what we see is hyperactivity, which is then followed by tissue degeneration and less activity. So any technique that would allow us to target this aberrant activity in the hippocampus in a non-invasive manner could have potential a lot of applications, both clinical, but also as a useful tool in neuroscience to target deep regions of the brain and explore them better. All right, so what I'm going to show you today and what we've been working over the past years is the translation of this technique to humans. And as you can imagine, this does not come without its challenges. I think the very first obvious one is a challenge of scale, right? And then we also need to think about safety. So in the mouse model, they could do suprathreshold, so they can actually elicit action potentials. It's not something that we're going to do in humans to start with, at least. The other challenge has to do with electric where you place the electrodes. So in the mouse, they put them directly in the skull. In humans, you're going to put this non-invasively in the scalp. The other has to do how do you do target verification, right? So here, they had to sacrifice the animal, look at staining in cells. That's not what we're going to do in humans either. All right. So throughout this talk, where I want to take you is in this journey where we are on how we go about translating this technique to humans. And for that, I would argue that we need at least 3 main ingredients. One has to do with target validation, so we need to show that we're targeting the hippocampus. The second has to do with physiological modulation, so can we change the physiology of this region? And the third has to do, can we change behavior? All right, so here in a nutshell, in this figure, you can see a schematic of temporal interference. And the idea here, simply put, is that you have these 2 current sources. So you have the green, okay, you can see the green one and the orange one over there. And what you do is that you use these 2 current sources at kilohertz frequencies, and they will be shifted by a small amount that is meaningful to the brain, okay? So typically, if you want to do techniques like TACS, you wouldn't be doing 2000 hertz. You'll be doing something between 1 hertz and 100 hertz. And here we want to target the hippocampus, so what we want to do is to target a delta F, a different frequency that we believe is meaningful to the hippocampus. So here we're going to do theta frequency. So we'll have one current source at 2000 hertz and the other at 2005. And then the idea is that these currents will mix, okay? They'll generate an interference pattern. Where the interference pattern is generated, you have an envelope, and it's the envelope that will modulate neural activity. In the bottom part, you can see the montage that we use. So we have our green and our orange electrodes, okay? Each color matches an electrode pair. So the very first thing that we do for target validation is to use a computational model. And so we have a final element model, very well described, lots of tissues. So what we started by doing here was to define one region of interest in the left hippocampus. That's the area we wanted to target. They're in pink. And then we define other ROIs, regions of interest. Those are the blue spheres that you can see underneath the electrodes and between the electrodes. And what we expect to see is a higher field envelope amplitude in the hippocampus compared to the cortex, okay? Because this field envelope, and I'll refer to it again and again, is this TI quantum, okay? It's what we want to quantify, it's the envelope that you see at the beginning of my slide. Okay, so this is what we see in the model, quite promising. We see higher envelope amplitude in the hippocampus compared to those blue spheres in the cortex. Okay. The other interesting thing about temporal interference, so when you put more than one current source in the brain, is that you might steer your current. So the whole idea here is that you keep the electrodes in the same place in the head, but you just play with the current ratio between, in this case, your orange and your green current sources, okay? And the idea is, if you do this, so let's say you start with same current ratio, 2 electrodes, you'll target something central between the electrodes. If you push, and I'll show you a representation, if you play with the current ratio, you should be able to push the locus, the focus of that envelope, more anterior or more posteriorly. So to validate that, to check that, what we did was we segment the hippocampus into thirds. And so there you see the anterior, mid, and posterior portions of the hippocampus. And again, we look at a model. We start by looking at this one-to-one. we see that the field envelope amplitude in the hippocampus pretty much, in this case, homogeneous. And then we do this 1 to 3, so 1 to 3 current ratio. What I'm doing here is that the green electrode now has 3 times more current than the orange electrode. And by doing this, what should happen is that you push the current, the envelope, more towards the orange, so the one with less current intensity. And that's what we see here. So we see more current intensity in the anterior part of the hippocampus. Okay, so that's nice and well, but it's a model, and we wanted to see what happens if we actually have a human head. We work with colleagues in Marseille that gave us access to a cadaver, and the advantage here is that you can implant the electrodes in the exact positions that you want to answer your questions. So the surgeon implanted the electrodes along the longitudinal axis of the hippocampus. You can see there the three electrodes. I think the pointer is losing battery. What we do here is, again, we measure the envelope amplitude. So if you're following me, this is your TI locus, so you're interested in seeing. We see that the envelope amplitude is higher in the hippocampus. You see the black outline there, and is, thank you very much, and is lower in the cortex underneath the electrodes. Whereas the absolute amplitude, or TACS, this will be equivalent to doing alternating current simulation, your amplitude is higher close to the electrodes compared to the hippocampus. And here you can see just a plot of this, one of the electrodes. See this one highlighted here with the white dot. So you can see that the electric potential that we can measure in the cadaver, in this electrode, what happens here is that with a normal simulation like TACS, you have a decrease in electric potential with depth, whereas with TI, you have an increase in electric potential with depth, which is what you want. And this is just a box representation of the same. And another way of looking at this, I think a nice way of looking at this visually is thinking about the envelope modulation ratio. So I told you about this two current sources, they're mixing, and what we hope to see is an envelope modulation. And this envelope should be at that difference frequency, in my case, 5 Hertz. And the more modulation you have, the more they are mixing. And you can see that you have an increase in the envelope modulation ratio with depth. And it's peaking where the hippocampus is. So up to here, I hope I've convinced you that we showed some target validation. So we showed that you have higher envelope amplitude in the hippocampus. And if you're looking at normal AC, this amplitude will be higher, so we'll peak in the cortex. Okay, so all in all, that's cool, but what happened, can we change physiology, okay? So in order to do that, non-invasive, in humans, we need a technique that will allow us to look at the hippocampus. So we got participants to go in a scanner, and we measure fMRI, so bolt signal, while we deliver stimulation. So we ask our participants to do a simple task. This is an associative memory task, so the hippocampus likes this type of tasks, and our participants were asked to do something that you've probably done quite a bit while you're here. So you see a face, and you need to recall someone's name, right? You see my face, my name, and hopefully later on, you'll remember that my face goes with my name. And during this period of we call encoding, we apply one of 3 stimulations. So either sham, so the current just ramp up or down and down. This current ratio 1:1, so we apply 2 milliamp per current source. Or this 3:1 ratio, where we apply 3 milliamps in the green pair and 1 milliamp in the orange, pushing the current to the anterior hippocampus. And one important thing here is we really only stimulated for 32 seconds, so very, very short. We're interested in see whether we can modulate the physiology in the absence of behavioral changes that can be a bit complicated to understand. All right, so the very first thing that we do is looking again at target validation. This time, because we had our participants in the scanner, we can make individualized models, so we can see whether this matches our original model that I showed at the beginning of the presentation. So I do the same thing. For each participant, I segment their hippocampus in thirds, and we look at the field envelope amplitude in the mid portion of the hippocampus and the anterior and the posterior. And the hopes is, if you do this one-to-one, you should target more the mid hippocampus, and this is what we see here. If you do 1-3, you should target more the anterior portion of the hippocampus, and this is what we see here as well. All right, so now let's look at what happened in terms of bold activity. So first I'm just showing you what happens in sham, so in the absence of simulation. I told you that we chose this task because it's hippocampal dependent, and we see a nice activation of the hippocampus when you're doing this encoding, binding of face and names. And we see that there's an increasing bold signal both in the left and in the right hippocampus. Is there during encoding but not during recall. And importantly, and we knew this a priori because there's literature on this task, we should see higher activity in the anterior compared to the mid and posterior. So it's a good task for us to put the simulation on top and test what happens when we stimulate with TI. Okay. So this is what we've seen. So when we apply this one-to-one, we still see activity in the hippocampus, very much like we had in sham. But when we apply the 1-3 during, we only apply the simulation during coding, we see a reduction in the bold signal, okay? And this is what I'm showing here. So in the left hippocampus, we see a reduction in bold signal when we apply 1-3 compared to 1-1 and sham, only during encoding, which is the only period that we apply stimulation. Then we also look at the right hippocampus, so on the other hemisphere where we didn't apply stimulation, and on the right hippocampus, we do not see changes in bold activity. We also look at the cortex underneath the electrodes, and here, again, as a control region, we do not see changes in bold activity. So I've showed you that we can change physiology, but then we ask, okay, we had a task where the hippocampus was engaged. What happens if there is no endogenous activity in the hippocampus? So we got a new set of participants. We did something very similar. They went in the scanner, we stimulated, but now during this task that is non-hippocampal dependent, we see that in sham there's no activity, no increased bold signal in the hippocampus, and we see that now when we stimulate, we do not modulate hippocampal activity. So very much what you would expect from a technique that is doing neuromodulation is working with the endogenous activity that you have. So finally, so we show this physiological modulation. We want to look at, can we modulate behavior? So we got a new set of participants, and they came for 2 sessions in the lab, and we selected the TI 1-3 condition where we saw an effect on bold and sham, and we asked them again to do this task, but now for a very long period of time. So our blocks are no longer, they don't have just 3 blocks with about 9 minutes. They're now going to do this for more or less half an hour, a little bit more. We included this tractor here between encode and recall, just makes sure that it's really hippocampal dependent, not working memory. They get now simulated throughout because we really wanted to accumulate this effect of stimulation. And what we see is that when we do this, participants get better when they receive TI compared to sham. And here, this is the 3 colors match the 3 responses that a participant could give. So in a situation like this, you can be correct. So you selected the right name. You can select a foil, and a foil is a name of someone that was in that block, so another of us but does not match the correct face, or a distractor, which is someone name that we didn't present before. And this just allows us to run a multinomial logistic regression. So we see that during TI they're better. I've also done this just separating by the binomial. So we have correct and incorrect. Incorrect is the sum of the foils and the distractors. And in the middle, we wanted to check, this was a Bayesian model, so we can give some credible intervals. And what we see is on average an increase in performance of about 12%. It's nice. And then we do not see changes in reaction time or confidence level. Another thing that we did here was that we stimulated and then we stopped the simulation for half an hour, and then we asked participants to recall all the pairs again. And at this point, we no longer see significant changes in recall. So we are seeing those during the stimulation, but not half an hour later. Okay, so just to finish, because I'm sure I've run out, I'm a bit over. So one thing that I want to summarize, so one thing that we also want to guys to bear in mind is out of the slides that I showed you, we, these 3 studies, we had about 58 participants. The TI simulation was really well tolerated. We didn't have any adverse effects. Any side effects were compared to other transcranial electrical simulation approaches. The thresholds at which people feel this are much higher. As I was telling you, we're pushing 3 milliamps, a 1.5 centimeter electrode. So this with TACS would have been really, really painful. We don't see this. The majority of our participants not feel the stimulation. And we also, in this long, when we did the behavioral, we probed participants between the task. We asked them, do you think you're having simulation? How confident you are? And we compared that to sham, and what we see is that they were effectively blinded, which is also very good. So all in all, I hope that you're excited and hope that you're convinced that we showed you target validation in silico, ex vivo, and in vivo, and that we can target the hippocampus in humans non-invasively, showing physiological modulation, so selectively targeting the hippocampus. In this case, we wanted to target the left hippocampus, and also having behavioral impact from this targeting. And yeah, just to acknowledge lots of people that worked on this project, and quite a few people from my group are here at the conference, and their posters are here, so if you have some time also do stop by those. Thank you very much.
Thanks a lot, Ines, for this exciting talk and this exciting data. So there are some questions in the Q&A, so I will read it to you. Do you foresee validation in epilepsy patients with hippocampal electrodes in the near future? What would you expect to see?
Yeah, for sure. Yes, yes. I think we just went to something, so future, we want this to be non-invasive. We also actually started a clinical trial based on this results to patients with Alzheimer's. But yes, I think it'll be really useful to have other validations. I know there are groups that are working, we'll apply it in the macaque, so we'll have additional validation from animal models as well.
Thank you. Next question, how large is the volume stimulated by TI as a function of depth? So it's kind of...
So, very good question. I don't have an exact answer for you. So in terms of millimeters, I was trying to go back, it's a long way. So you can see that the region, it will be along this path. We didn't have an electrode here to record. So of course, we're not at a point where you could target, at least with just this montage that has 2 current sources, that you could target, for example, a nucleus in the basal ganglia. So we're targeting a large volume along the hippocampus. I think, and you could improve this from doing, from putting more current sources. So you can make the locus even more specific. So yeah, it's focal, but it can be made even more focal.
Thank you. I guess we have to probably move on, and if at the end is time, we can get more questions. Thanks a lot again, Ines. So the next presentation will be by Elena Beanato. She's currently performing her PhD at EPFL, and she will show you data about striatal temporal interference stimulation and behavioral and imaging impact. Elena, the floor is yours.
Yeah, thank you very much. So yes, I will now continue and will focus with the second talk about modulation of the striatal activity as mentioned with the same technique, temporal interference. So we now switch to motor learning instead of cognitive, and why we do this, it's actually because we know that motor learning is one of the key domains we use in our everyday life. So if you imagine how to learn to use a new tools or play an instrument. And in addition, nowadays population is aging, and with age we have several neurodegenerative and neurological disorders that are appearing mostly. And just one of the examples is about stroke, to give you an idea, and here the incidence is increasing in older people. In this case, several patients remain with impairments after the accident, in particular motor impairments. These are just some of the reasons why motor learning has been highly studied already. And to give you an idea, we start by looking at the brain regions that are involved in the motor learning process. And here in this meta-analysis, you can nicely see how we need an interplay of different regions within the brain, and both cortical and subcortical regions are highly important. And in between the subcortical regions, one of the key hubs is actually the striatum. So this is why the target region we will target with temporal interference in this project will be the striatum. Again, since motor learning is highly important and the striatum is an important region for this process, also we have already some knowledge about the function and the structure of the striatum. And this region can be subdivided into three main parts based on anatomy and functions. And we have, from the ventral to the dorsal part, a limbic, an associative, and a sensory motor part. These different subregions are differently involved in the motor learning process. Here you have a scheme about the motor learning, how it evolves with time. And in this project, we will mainly focus on the first part, so the fast learning. So when we start to acquire new skills and we fastly improve. And in this fast learning, it was observed in both animals and human work that there is actually a shift of activity from more associative to sensory motor part of the striatum, naturally coming with practice. So we now have our main domain we want to target, motor learning. We have our main structure we want to target, the striatum, and how to actually modulate its activity. So the simulation protocol we decided to use, it's a well-established protocol in the TMS field, which is intermittent theta burst stimulation. So from an animal study, theta burst stimulation was shown to be a plasticity-inducing protocol with a plasticity, long-term potentiation, and long-term depression effects. And in particular, we will use intermittent theta burst stimulation, which consists of applying 2 seconds of stimulation followed by 8 seconds of pause, and this repeated one after the other. And during these 2 seconds of stimulation, we will apply 10 bursts at 5 hertz, and each of these bursts will be composed of 3 pulses at 100 hertz. So here you can nicely see how we produce this with the temporal interference concept. So again, you have the 2 signals, the 2 high-frequency currents that, by combining the 2, can form different protocols. In our project, we created 2 main conditions. So the first one, which is our control stimulation, which we will call HF control, it's high-frequency control, meaning that we won't apply any shift in between the 2 currents, so the envelope will be flat. And for our active condition, for the stimulation, we'll, as mentioned, mimic the intermittent theta burst pattern, and here you can maybe even nicely see the description that I gave you before, so the 2 seconds of train followed by 1 second of break, and then each of these 2 seconds, the 10 bursts, composed of the 3 pulses, each of them, of each. And we now have the basis for our hypothesis. So what we wanted to test was if we can, with intermittent theta burst TI stimulation, we will be able to reach and modulate the ongoing task-induced brain activity within the striatum, and by doing this, also to modulate behavioral outcomes in humans. A little bit of a spoiler about our findings. So we have three main results. So with ITBS-TI stimulation, we could actually find a differential pattern of local brain activation within the target structure, the striatum. We also observe an increased bold activity within the whole motor network. And finally, these brain effects reflected into an improvement of motor performances. Let's start with the first result and how we arrived to that. In order to test this, we performed an MRI study in which we asked 15 young healthy subjects to undergo a crossover randomized double-blind design. And in the first part of this protocol, we asked them to perform 2 sessions of resting state, because the first question we had was already if we could see a modulation whilst the subjects were at rest. In this case, we applied either the active or the control stimulation in each of the session in a randomized order, and in each session, we asked them to perform 8 minutes scan before, during, and after the stimulation. What we found after the processing tried to limit artifacts for movement and electrical currents, we applied a seed-based connectivity analysis with the target in the right striatum, and we observed actually no significant difference between the two conditions as expected. So we then do a further step with the second part of the protocol, in which we ask the subjects to perform a task-based fMRI, again in a crossover design, so 2 sessions, with either active or control stimulation. And in this session, we ask them to perform the motor learning task that we choose, which is the sequential finger tapping task, which is a task that is highly used in this field. In this case, the participants had a sequence of digits in front of them, and the goal was to reproduce this sequence by pressing on a 4-button box, with each finger corresponding to a specific digit. The sequence was shown during the whole duration of the task in order to reduce the working memory load, and this was done over an hour of scan. The protocol consists of, firstly, a baseline of 30 seconds without acquisition of images in order to have the individual performances before starting the protocol, and then they perform 6 big blocks, with each block lasting around 9 minutes and a half, with 10 repetitions of the task with 30 seconds block intermingled with 30 seconds of break. And this, again, twice, with at least 3 days in between the sessions in order to have a washout period. Then the first question we had was about the target area, if we could actually observe changes associated to the stimulation. And here we analyze firstly 2 subparts of the striatum, which are the putamen and the caudate. And here you can already nicely see that in between the 2 structures, the task was engaging the putamen more than the caudate region. And actually, in the region in which the activity was already higher, we had an effect of the stimulation, which increased even more the activity associated to the task. This is actually pointing toward the fact that the stimulation effects are specific to the subregion in which the endogenous activity is already higher, as already shown in the previous talk. And then we wanted to do again a step forward. I mentioned this natural shift that happens during the motor learning process. And so we looked at the subparts of the striatum which were showing a linear increase or decrease of activity along time. And what we found, here you can see for both stimulation, different slices of the brain. And in hot colors, you have voxels that show increased activity along time, and cold colors a decrease. And what we observe is actually in accordance with the literature, with an increase in the inferior part, which is more the sensory motor part of the striatum, and a decrease in the superior part. And in order to even look better at this shift, we also plotted the peak activation following time. So here you have one slice per block. So on the horizontal axis, you will have time as measure. And from the beginning of the task toward the end, we actually see that ITBS-TI, we can observe a shift from the most superior to most inferior part of the striatum, whilst for the control, actually the position of the peak is quite stable in the superior part, meaning that this natural shift could be supported by the TI stimulation we applied in order to fasten this shift. Okay, so here was about our target region, but then we also wanted to look a little bit what was happening at the whole brain level. So this is what we did. And we started by looking at the activity induced by the task, so during the control stimulation. And here you can nicely see the motor network, which is activated by the task with primary motor regions on the right side, since the subjects were doing the task with the left hand, and secondary region also in the ipsilateral side. And starting from this, we then compared the two sessions between ITBS and HF control on the activity level, and we see that the contrast, there is a significant higher activation during ITBS with respect to HF control when inside the motor network, sorry. And here you can see different regions already. Our striatal region, which was expected since we already saw the bold activity plotted with an increase in the putamen, and also other regions such as the thalamus, the supplementary motor cortex, cerebellum, etc. And finally, all of these, actually the goal would be also to have behavioral effect, not just at the brain level. And in order to do so, we then investigated how the stimulation was acting on the motor performances. To do so, we actually did a step, let's say, back, so outside the MRI scanner, and we asked a new set of subjects, again 15 subjects, to undergo a sequential finger tapping task outside the scanner. The task is exactly the same with the sequence, just this time we had longer blocks with 90 seconds, again, one block of baseline before starting, and then in a crossover design, 7 blocks of 19 seconds intermingled with 90 seconds of break. And just as a notice, in the results I will show, there will be just 6 time points, just because the 4th block was a random sequence that we excluded for now in the main results. This in total, with respect to the MRI protocol, which was lasting 1 hour of training, here we had 20 minutes of training, so a shorter protocol. What we actually found is that by investigating 15 healthy young subjects, here in this case we didn't see any behavioral effect. But then we repeated the same protocol on an older population, again 15 older subjects, and in this case what we observe is an increased performance during the ITBS-TI stimulation with respect to control, which indicates that this stimulation can be beneficial at the behavioral effect. And here, the improvement that we observe, it's actually up to 33% with respect to the control. This is, again, a little bit what we expect, because we already know that in the older population, we can have different hypotheses, but already the performances are lower than in the young population, so we can imagine that there is a wider range of possible improvements. But we also know that, for example, brain plasticity is lowering with age, and so we could think that with the stimulation we could push this plasticity toward a more, let's say, younger state. And then finally, here are the results from the outside scanner, but we also had data from the inside scanner with longer, actually, training and stimulation time. And so what we observe is actually that we have an effect of the stimulation when the task was done for up to an hour of practice, again with ITBS leading to increased performances with respect to HF control. This points toward the fact that the dose could be an important factor for the stimulation effects. So in order to summarize all the results I presented to you today, we found that ITBS-TI stimulation led already to a differential pattern within the striatum with the putamen region showing higher activation with respect to caudate and with the effects on this region that was already activated. And also we saw the supporting of the shift, the natural shift that we have from associated to sensory motor areas with the stimulation. We then observe also an increase in the bold activity within the whole motor network. And finally, this change is reflected in behavioral outcome with increased performances during the motor learning. Thank you very much to everyone, but in particular to the Professor Hummel and the whole TI team that greatly helped the project. Thank you very much.
Elena, thanks a lot for this excellent talk. There are some questions already, and please feel free to add more. So one question was about your statement that cortical stimulation it's about cortical stimulation often change activity connectivity at rest. So why do you expect that TI only show modulatory effects during a task-dependent situation rather in the resting situation in your target region?
Yeah. So actually, it's a little bit in line with what Ines already presented a little bit before, but it's in this case, we are stimulating with subthreshold intensity, and it's really a neuromodulation which is not enough. It's not like TMS in which we induce firing, but we are really trying to support the endogenous activity that it's already present. So by just applying this subthreshold, we don't expect significant results because the region is not already activated, and then stimulation is actually probably not enough to induce any effect. But when this region is already activated, this means that the activity, or at least the activity is already a little bit higher. So by inducing even a smaller effect, we can actually push this activity even higher, let's say like this. But I think in the brain stimulation in general, when we apply electrical stimulation, we always try to combine these two in order to have a higher effect. But,
thank you. There's one question in regard of populations with neurological disorders like stroke, etc., whether or not it is applied already, and what is your kind of vision in regard of this?
So actually, we already started a little bit with some populations. So again, I mentioned stroke, and we are planning to apply this on a stroke population, but also, for example, in a TBI patient which had a traumatic brain injury, in order to always see whether we can actually act on the motor rehabilitation, which will be the final goal. And of course, there are other populations that are also probably suitable, such as Parkinson, for example, but this will be in the next step for sure. The more population, the more proof we have, the best it is, probably.
Probably one last question in the view of time, so I try to bring two questions together. So it's mentioned that ITBS for TMS shows quite a bit of inter-individual variability. And the question is, in regard of his effectiveness, if you see the same in TI, and then in this regard, whether a cohort of 15 or 20, like Ines showed, if this is enough, or what do you think? What is your comment about this?
So actually, I think this is also one of the reasons, for example, in which in young, during the short protocol, we don't see any effects, while in the older population, yes, because I also imagine if we increase the sample size already, we could observe more effects already in the younger population. And we observe, as I think in TMS and the other non-invasive brain stimulation, we observe some subjects that respond and some subjects in which actually the stimulation is not beneficial. So the effects we see is really on the group level. And the goal would be for sure to have a bigger population for the future. But I will say this was really a proof of concept study that we wanted to already test whether the parameters we were using, they were good, let's say like this, to target the striatum, because, I mean, here I didn't mention all the parameters, but we had a specific intensity, also for the modeling, as Ines mentioned before, the positioning of the electrodes. If we would have started already with a big population, it would have been possible that our parameters were simply not already optimized. So, but it's something
to go with.
Thanks again, Elena. There are much more questions, but probably at the end, if we have time, we will address them there. Thanks a lot for this fantastic performance. So now for the last talk in the session, I would like to invite Yuk-Jun Moon from EPFL, and he will talk about the impact of MTL, targeted TI stimulation, spatial navigation, in regard also of grid cell-like representation. Jun, the floor is yours.
Yeah, thank you, and thank you for coming for the talk. Actually, I was doing my PhD at EPFL, and at the very last stage of my thesis, so I had luckily had chance to apply the novel TI stimulation to my task. So yeah, it's my great pleasure to present this preliminary result here on behalf of the entire team. So the title is Impact of MTL targeted TI stimulation on spatial navigation, and especially I've used fMRI-based grid cell-like representation during my thesis. So so most of the imaging data is related to this part. It's not working. Okay, yeah. So yeah, why we target the spatial navigation? So like when you arrive at the Glasgow airport and come to this conference hall, so we always had to navigate to some areas, and then during the navigation we should always decide where to move or which way to go, and so but it's very important for our daily life, but unfortunately, it's well known that the ability of spatial navigation decreased as we aged. So yeah, I think we could try to enhance the spatial navigation ability using the TI stimulation. It's not working? Okay. Okay, so it's already well known that so many brain lesions are involved in the spatial navigation. But like what Ines did, so we tried to focus on the hippocampal, but also entorhinal complex, as it's really well known as a spatial navigation core. And especially, I would introduce grid cells, which mostly found in the entorhinal cortex, also in humans. So, yeah, the video doesn't play, but yeah. Anyway, so, okay, thanks. So, as you can see in this video, so while rodents are freely moving in this box, so the cell generated at very specific locations in the environment. So people consider these cells in called some allocentric spatial location in the map. And this is a kind of representing our spatial representation in our brain. And yeah, so many studies for these grid cells already demonstrated is deeply involved in the spatial navigation as well as the memory, and also the entorhinal cortex, where the cells are mostly discovered, is also very mostly affected the brain lesions in the AD, so Alzheimer's disease in the elderly. Okay. Okay. So as actually, unlike the rodent, as the actually single unit recording is quite limited in humans, so we use the fMRI-based estimation of grid cell-like activity. So basically, the idea is that so when the subject is navigated toward the grid or line direction indicated as in these orange arrows, So we expect a higher firing rate from the cells, while the lower firing rate when they navigate through grid misaligned directions. So actually, Dweller and his colleague earlier in 2010 expected that if they plot the bulb activity in the entorhinal cortex according to the navigation direction, then they expect the same hexadirectional modulation of the bulb activity in the lesion, and they call it as GCLR, grid cell-like representations. So so not only we can estimate the activity of the grid cell like population in those regions, we can also compare the activity between the condition. For example, so if in condition A, for example, if the fluctuation has higher amplitude, we can say this probably has a higher grid cell-like activity than condition B, which has a lower level of fluctuations amplitude. And for this presentation, just for the simplicity, I only show you the amplitude value, not the entire fluctuation shape. And this is there are more and more consensus that this represents some fMRI proxy of grid cell-like activity. Yeah, and fortunately, in our previous study, during the spatial navigation, we could also detect the this very characterized hexadirectional modulation pattern. So we adopted the same spatial navigation task in this study with small adaptation to fit the TI stimulation. And basically, the task is very typical one. So during the encoding phase, they had to navigate inside the arena to actually encode the locations of the object. And then during the retriever phase, so when the cued object appear in the screen, then they had to retrieve where was the where the object was in the encoding session, and then they had to navigate back to the original location. And then they repeat this for the fixed amount of time of 9 minutes. Yeah, maybe I won't really go into too much detail about the TI stimulation, as it's been already well introduced by two previous speakers. But yeah, just to mention about the MTL part. So yeah, it's been there's been lots of study try to target the MTL, especially hippocampus, but it was really challenging. So most of previous work saw TMS study has been indirectly stimulate the lesions by targeting the parietal region, which has functionally connected to hippocampus. But so with the TI stimulation, we could actually directly stimulate the hippocampal entorhinal complex, non-invasively, but selectively. So yeah, this is also very similar to the slide Elena already presented. So with the ITBS stimulation, with the 8 seconds of actual idle time after 2 seconds of stimulation. So yeah, this condition was suggested to have a more excitatory effect. And in addition to ITBS, here we also include the TBS, CTBS conditions, a continuous TBS condition, so which without any idling time, and suggests to have more inhibitory effect. So combining the three stimulation condition with the spatial navigation task. So we have experimental design like this. So we have total 6 blocks, and 2 blocks were actually stimulated with for each stimulation condition, and the order was still randomized. So during this, through this experiment, we expected a change in spatial navigation behavior, and hopefully to the improvement side. And of course, we also expected altered brain activity, especially crystal activity, and also the more broad spatial navigation network. So I first introduced the behavioral result. Yeah, good thing is that we could find the significant effect of stimulation on trial time. So this is a normalized trial time. And as you can see, so in TBS, ITBS conditions, so the trial time was shorter compared than the CTBS condition. So it means they were better in ITBS condition. So to further take a look at how this shorter trial time originate from, so we check the further behavior parameters, and then we found out that so while actually the actual moving time was more or less the same, like this, but so deviation ratio, so basically it's calculated based on the actual moving time over the trial time was a bit higher in the ITBS condition than the CTBS condition. So they more tend to move during the ITBS condition than the CTBS, later being stationary. So this may suggest some faster decision on where to move during the ITBS condition compared to the CTBS condition. So we also checked another behavioral performance, for example, the distance error, so which is that so the distance between the correct object location and record location, and we couldn't find any significant difference in there, and also total navigated distance was also in the same level across the condition. So it suggests that so there was no actually trade-off between the shorter trial time and the other kind of behavioral performances. So yeah, I would summarize the behavioral result that so we could observe the impact of TIS on spatial navigation. Specifically, it could reduce the trial time in ITBS compared to CTBS condition. And this suggests improve the spatial navigation in terms of time. And this shorter trial time and higher navigation ratio in ITBS may also, or alternatively, may also suggest some possible facilitation of tendency or impulse to navigate rather than to be stationary. Also, we take a look at the fMRI images to see the associated brain activity changes. And yeah, I would introduce the crystal-like representation result in the right entorhinal cortex, where we stimulated through TI. And as you can see, so compared to the control condition, so crystal-like activity was decreased in both ITBS and CTBS condition. And although actually the statistically it was not significant, but it seems like ITBS has actually a higher impact of the stimulation than the CTBS. And we also checked the other four symmetry, 4-4, 5-4, 6, 7-4, and then so we confirmed this. So this stimulation dependent change was only observed for the 6-4, so crystal-like representation, and not the other control symmetries. And also, interestingly, this impact of TIS on crystal-like activity was actually observed in bilateral internal cortex. We only stimulated right side, but yeah, the effect was only observed in the left side as well. So also to find some brain activity which can explain the behavioral changes, a shorter trial time. So we assess the correlation between the beta in the right hippocampus area and the trial time, and we could find a very significant correlation between the beta changes between the ITBS and CTBS and trial time changes in the same condition pair. And this was also the same for the navigation ratio. So it suggests that the bigger the hippocampal activity changes during ITBS than CTBS, then we could expect the faster trial time during ITBS compared to the CTBS. So to summarize, so TI stimulation on hippocampal internal complex modulates spatial navigation behavior. So we could observe the reduced trial time while increased navigation ratio, which is in total suggests the improved navigation performance. And TIS attenuated Gracie-like activity in the internal cortex. And also TI stimulation associated right hippocampal activity increase could explain the reduced trial time during the ITBS. So yeah, I would say this showed potential future avenue for navigation enhancement in humans. And I would like to thank the entire team and include Professor Olaf Blanke, who actually couldn't come here today due to the flight issue, but yeah, thank you very much.
Jun, thanks a lot for this excellent talk. So I think we have still around the 10, 11 minutes for questions. So probably maybe you join here also on the board, and then we can go through the different questions. So first question for you, Jun, probably is, so there's one question about your CTBS effect and which whether or not it impairs basically the spatial navigation performance. If you could comment on this.
Okay, for the, I don't know, the negative effect was at least not significantly observed. So yeah, so there were actually lots of subject-wise variance. So yeah, statistically, yeah, it was not significant at least.
Okay, so one question which popped up a couple of times for Ines basically is, so you showed a reduction basically in the hippocampal bold activity, but on the other hand, there's behavioral improvement, right? So could you comment on this, how you bring this together, what you think, what is the underlying mechanistic explanation for it?
Okay. Yeah, sure. So when we started, we didn't have a priori hypothesis whether we would see an increase or decrease in bold signal. We do see that it decreased. Interestingly, I didn't have time to show all the data, but what you expect is when you are on your correct trials, so the one you're able to encode, versus in your incorrect trials, you have increased bold signal. And we see this in sham, so correct is higher than incorrect. And we see this particularly in the anterior portion of the hippocampus. When we do that 1-to-3 current ratio, we see the same. Correct is still higher than incorrect, but there's a shift, okay? How can I explain this? As you know, it's really hard to explain how bold relates to spike timing or relates with synchrony. It is possible that we do have a decrease in metabolic demands, but the process is still the same, okay? So you still have correct showing high bold signal than incorrect. Then you asked me about how do we go about relating this with behavior? And that's an awesome question, and I'm afraid I only have a partial answer to that. Because in the fMRI study, we stimulated just during encoding. And when we did the behavioral study, we made a decision to stimulate for much longer. So it is possible that the behavioral effects that we are seeing do not come necessarily from the modulation of the signal that we saw in the encoding, but could come from either the distractor part or the recall. And this is something that we very much would like to investigate, but something that we would like to investigate with future studies.
Thank you. And then there's a question for Elena. What's the spatial resolution of the stimulation, and can you specifically target the sensory motor parts of the putamen?
Yeah, thank you already. It's a great question, but it's a little bit in line with the results we show. So the spatial resolution, we are talking about centimeters. So we actually perform the same optimization thanks to our collaborators, in which we try to optimize the accuracy of the stimulation. But the main goal was to be able to reach the structure without the overlying structures, but also to combine this with the activity. So this is a little bit the same as saying we were not targeting specifically the putamen, but since the putamen was already engaged in the task, we could observe the effect. So even though we were targeting the whole striatum, both associative, sensory motor, and a little bit the limbic part, the goal was really just to be able to target structures that were already engaged. So,
yeah. Can I just, so I think it is with any other of these electrical stimulation techniques, you need to think of the fact that you will have a distribution of current per area, right? So it's about what is enough to modulate the area that you're targeting. And this is something that is tricky, not just for this, but to others as well. So it is just a complex question, because we need to think about what we're talking. Are we talking about what are the areas under the threshold? Are we thinking what is the current density for the area that we target? And yeah, just hopefully confusing but clarifying.
Thank you. Probably a question for all of you, and you can just decide who want to start to answer it. So how are you able to reach a deep brain when performing interference stimulation using 3 milliamps? While models show you would need higher currents around 8 milliamps, what is the effect of shunting of your current over the skin and thus stimulation of peripheral nerves?
Yeah, I can start. So I think if I'm not mistaken, that paper talks about suprathreshold. So what would you need to actually generate action potentials? That's not what we're doing here.
No, I think it's enough. It's clear. Okay. So let me see what else we have. So maybe for June a question. 1 second. So did your paradigm allow you to look at effects of learning rate, drop error over time? For example, that indicates, did the different stimulations affect performance over time, learning rate differentially?
Yes, so of course I could take a look at their learning curve, but so unfortunately this task was designed to be a bit easier, but so it's so yeah, there was not significant difference in the running rate between the conditions, and usually it's a bit hard to observe even for my another studies. So most of the actually learning happened in the first block, and then so their performance more or less saturated after the second one. So yeah.
Thank you. Probably I summarize 2 questions because they are questions, I guess they are for all of you. So one question is, so how do you basically select your beat frequency? Why theta bursts? Why theta stimulation? And the other one is whether or not the effects persist much longer after the stimulation. I think you can all comment on this.
Sure. So Yeah, we have the trouble that you want to target a deep region in the brain non-invasively, so we don't have a readout for it, right? So what you would like to have is what is the frequency at the hippocampus or entorhinal cortex or the putamen. We don't have that. So at the moment, all these studies were based on literature, right? And we can think about how you could optimize that further if you have more information or if you have a cortical readout that maps the activity of a deep area that you're interested on.
Yeah, in our case, I would say we actually went to the theta burst protocol because we really wanted to induce this long-term potentiation depression effects, and we really wanted to, let's say, go a little bit risky, but also on the safe side in the sense that of choosing a specific stimulation frequency and target specific region sometimes because exactly the difference between subjects, the specific frequency could be individual and this could actually affect the results. So with at least the TBS protocol, we were really trying to target this kind of plasticity. And maybe also to add for the behavioral effects over time, I didn't mention before, but in the protocol we also had follow-ups right after the simulation, 90 minutes after and 24 hours after. And in this case, we didn't, at least in our case for the motor task, we didn't observe any differences in between the stimulations. So the effects were really limited to the training time.
Yes, same with the results that I show you. So we don't know. We hope that it can be optimized. Like as you are seeing, we show you sort of proof of concept in our first applications. I'm confident that we'll be able to optimize to being able to have effects that outlast the stimulation, whether they would come out of longer stimulation sessions, higher current intensities is something that we absolutely need to explore, or cumulative effects. So in this case, it's just one session. But, you know, for example, for a TMS protocol in clinic, you'll do this over 10 days.
So yeah, thanks a lot. We have a lot of more questions, but we're running out of time. So feel free to get in touch with the speakers by email or whatever means we have, right. So thanks a lot for the excellent talks and your excellent answers to the questions. And thanks a lot for coming this early in the morning and asking so many questions. I hope you're also convinced that temporal interference stimulation is a new, very interesting method to target deeper structures, for using it for behavioral changes, but also to better understand the functional role in a more causal way of these deep structures in humans, like basic ganglia structures, hippocampus, or you can think about several other structures. So again, thanks a lot to all of you, and have a nice day. Bye-bye.